Hello, and welcome back to Human Evolution. Today I want to talk about DNA and RNA. First, though, I want to remind you about genes. Remember that genes are short sections of DNA. They can be up to several thousand base pairs. They code for proteins. These are the instruction manuals, and you have a specific section of each piece of DNA that will hold genes. And remember that alleles are different versions of genes, and we get the banding pattern on chromosomes based on where these genes are, thinking about euchromatin and heterochromatin. So when we're thinking about DNA, genes, and chromosomes, what is that relationship? A chromosome contains hundreds of genes, which are composed of DNA. A chromosome contains hundreds of genes, which are composed of proteins. A gene is composed of DNA, but there's no relationship to chromosomes. A gene contains hundreds of chromosomes, which are composed of DNA, or a gene contains hundreds of chromosomes, which are composed of protein. Well, we know that a chromosome is a lot of DNA all packaged up together, all coiled up, right? We know that those chromosomes have hundreds of genes on them, and those genes are composed of DNA. So number one, proteins will come into play because those genes code for proteins. So chromosomes contain genes, which are composed of DNA. And genotypes are the genetic composition of an organism. This is what you can get, right? Phenotypes are the expressions of genes. These are the physical traits. This is what you actually get. And we'll be using the terms genotype and phenotype a lot in the coming units. All right, now before we start talking about how do we go from DNA to proteins, we need to talk a little bit about RNA, because RNA is at least in our story of DNA to protein, is the messenger. It's the way by which we make proteins. It does all kinds of jobs. So we need to really specify what's going on with RNA. We know that RNA is our other nucleic acid. It's ribonucleic acid, unlike DNA, which was deoxyribonucleic acid. We know that the difference between DNA and RNA is in the sugar. Okay, we know that both DNA and RNA are comprised of nucleotides. So when we look at DNA, we can see it is double-stranded, D, double, right? RNA, though, is single-stranded. It's always single-stranded, and that means its little nucleotides are open and can bind with other little nucleotides. And that's the key to RNA, is that it is active. It can affect changes in things, whereas DNA has to be opened in order to be read. RNA is always open. Okay, so which is the correct comparison of DNA with RNA? DNA and RNA have different purines and different pyrimidines. DNA and RNA have the same purines but different pyrimidines. DNA and RNA have the same purines and pyrimidines. DNA and RNA have different purines but the same pyrimidines. So let's go back one slide and look at this, and we're going to come back to this question. If you look, so remember, cut the pie. We said it was CT the PY. It's really CUT the PY, cut the pie. Our pyrimidines are cytosine, thymine, and uracil. Uracil is exclusively an RNA nucleotide. So when you're looking at the basic structures of those functional groups of the nucleotides, DNA and RNA share cytosine, guanine, adenine. But DNA has thymine, and RNA has uracil. Now, having said that, they don't really share exactly any of the nucleotides, right? They have the same functional groups, but remember they have different sugars. So an RNA cytosine is different than a DNA cytosine. And so if I were to say, well, how many nitrogenous bases are there? Well, there's cytosine, guanine, adenine, thymine, and uracil. There are 5. But if I were to say, how many nucleic acids are there, right? How many nucleotides, sorry, not nucleic acids, how many nucleotides are there? There are in fact 8, because remember, this cytosine in RNA is bound to a ribose sugar. This cytosine for DNA is bound to a deoxyribose. That's their sugar, right? And so there are 8 nucleotides, but there are 5 nitrogenous bases. Okay, so going back to our question, it's a little bit more complicated. DNA and RNA have the same purines, right, because the purine is the nitrogenous base, but they have different pyrimidines because of that thymine and uracil, okay? They have different nucleotides. They have similar, or they have the same purines, nitrogenous bases, but different pyrimidines, okay? So when you look at that binding, right, that base pairing during DNA replication, right, you can see when DNA binds, DNA binding to itself is going to bind GC-CG, right, AT-TA. But in RNA, there is no T, there's no thymine, it's uracil. So when you have a DNA-RNA bind, which is relatively unstable, by the way, and we'll talk about that more later on, you're still going to have the G and the C because those nitrogenous bases are the same. You're still going to have the A to the T because the T is in DNA. But the uracil in RNA takes the place of thymine, and so you get UA bonding here. Right? So it'll be RNA, the A in RNA will bind to the T in DNA, but the uracil, the U, will bind to the A in DNA. Okay, so which of the following nucleotide chains could be part of a molecule of RNA? Well, RNA does not have thymine, so the only option here is number 2, because there's your uracil right there. All right, RNA is probably the first genetic information. It's actually, interestingly, much more active than DNA. It does all kinds of things. Not only can it store information, and it also codes for proteins, right? Because it's reflective of the DNA sequence. So it synthesizes proteins, which is that whole like life function thing, but it can catalyze all kinds of cellular activities. So RNA is really, really powerful. It does lots and lots of things, and you actually are already familiar with another way in which RNA behaves, even though you probably don't realize it, and that is a cellular component you already know that is made of RNA coupled with structural proteins. What is that? Well, of course, it's your friend the ribosome. Ribosomes were not membrane-bound organelles. They were RNA coupled with proteins that synthesize other proteins. Okay, so there's lots of types of RNA, actually, and the ones that you're going to need to know are the messenger RNA, the transfer RNA, and the ribosomal RNA, and you already actually know about ribosomal RNA. Now, I do want to point out that when I talked about DNA replication, I did mention that there was an RNA primer in there. So you've got RNA that acts as primers for replication process. Also, you're going to have little RNA primers in other places too. There's also the ribozymes. These are RNA enzymes. And when we're talking about splicing in this conversation, removing introns, we're actually talking about a different little RNA machine that does that. And then there's other RNA that we use in all kinds of things. RNA can be used to regulate genetic expression, which we'll talk about at the end. But again, what I want you to know is about messenger RNA, transfer RNA, ribosomal RNA, and choose one or two of these other functions to be familiar with. So now that we understand RNA better, we can talk about the central dogma of molecular biology, and this is that cascade from from DNA to protein. So our genes get transcribed into RNA, which then get translated into proteins. And today we want to talk about this transcription process. So when we talk about transcription, we're talking about RNA synthesis. This is the process by which a molecule of DNA is copied into a complementary strand of RNA. Okay, it's actually called DNA-directed RNA synthesis. So the key player in this, though, is going to be RNA polymerase. When we were talking about replication, we said there was a little protein polymerase that made DNA. That was DNA polymerase. Well, in transcription, we don't want to make DNA. We want to make RNA. So we have an RNA polymerase that specializes in making RNA. There are 3 stages to transcription. There's the initiation, the elongation, and the termination. Okay, but in a nutshell, what happens, and I'll talk about each of these stages in just a second. You have to unwind a section of DNA. Now, because it's different. You're just going to make what's called the transcription bubble. You only want to open the part that you are going to actually transcribe. So you unwind it, and then it has to be rewound. Now, the coding strand is the gene, right? The coding strand is the gene, and you need the RNA to match the gene. And what that means is that you have to use the other side of the DNA, which is called the template strand, to make the RNA. So we want the RNA to match the gene, so we use the complementary strand to make the RNA. Okay, so we've got the template strand. This is where your RNA-DNA hybrid will be while it's being produced. And then you have the coding strand. This is the actual instructions. Okay, so when you're looking at this very complex diagram, you've got DNA and RNA, and then you have your little ribosome situation. This is a diagram of all of that central dogma we were talking about, DNA to RNA to proteins. But in this, which molecule is coded directly from DNA? Well, you can tell by the U's and the single strand that this is our RNA strand. It will be coded from the DNA. This will occur in the nucleus. Then it will leave the nucleus and will be transcribed into a protein. And that we will talk about later on, this exiting and being translated. Sorry, I said transcribed, I meant translated. It'll be translated into a protein. Okay, but one is the mRNA. And that is what we are making with transcription. So in initiation, there is a promoter sequence that some transcription factors called the transcription complex will recognize. They will bind to the promoter. Once they bind to the promoter, they will attract the RNA polymerase. The RNA polymerase will bind to the strand. Within the RNA polymerase, it will open it, create the transcription bubble, and it will allow for the RNA nucleotides to come in. They will attach to the DNA strand in the appropriate complementary order, and they're going to be on that template strand. And then, right, which is the elongation part, and we'll talk about that in just a second. But this is the initiation, the opening of the transcription bubble by the RNA polymerase, which bound to transcription factors, which knew where to go because there was a promoter sequence in the DNA. Okay? Now, in elongation, as soon as the RNA polymerase sits, opens up the DNA, it will zip off the transcription factors down the DNA, opening that bubble as it goes. The direction of transcription is called downstream. It will follow along that template strand of DNA. The little RNA nucleotides will pop on, and then as it travels, though, the tail of the newly made RNA will come out the back of the polymerase. And there it goes. Now, you'll see you're on that 3' to 5' side. We're growing on the 3' end, just like we always do. But what you have here is the coding strand is 5' to 3'. So the template strand is going to be transcribed 3' to 5'. This is similar to the direction we were going in DNA replication, but we're actually reading, right? We're reading this way, 5' to 3'. Okay, so this is elongation. It's going to continue to do this. The RNA is going to come out the back until it reaches the termination sequence. So you've got your coding region. This is what's going to be coding along elongation. And then you reach your termination site. The sequence in the termination site destabilizes the polymerase, and the polymerase pops off the strand and then goes off to transcribe something else. And what you get is this RNA transcript. Now, it's not mRNA yet. What we need to do is we need to make this RNA transcript, this pre-mRNA, into a message that we can take the information from the nucleus into the cytoplasm. That is the whole point of mRNA. Okay? Now, before we talk more about that, let's talk a little bit about which of the following is not a product of transcription. You do not get DNA from transcription. You do not get DNA from transcription. You get all RNAs from transcription, no DNA. Okay. Now, I'm going to show you a picture now which sort of puts everything together, and we're going to come back to it several times throughout our conversation about DNA to RNA to proteins, okay? But basically, all of your RNA genes and your protein genes are in the DNA. They are all transcribed, okay? Ribosomes are made of RNA with some proteins. mRNA, the messenger RNA, is what is going to carry all of the instructions. And then tRNA is something we'll talk about when we talk about translation. Okay, now I told you that this messenger RNA, right, that transcript that we just got through transcription is not actually done yet. Messenger RNA, remember, is going to be the gene instructions, so it can be made into a protein. It's called a messenger because it carries the message, the information from the nucleus to the cytoplasm, right? DNA never leaves the nucleus. Proteins are made in the cytoplasm where the ribosomes are. So the messenger RNA makes the journey between the two, okay? Now, this primary transcript has to be made into a mature transcript, and there are 3 things that happen to make that true. One, as it's being transcribed, it gets a whole bunch of adenines put on its 3' end. At the end of it, this is the last thing that happens. This really long poly A tail, it's just a whole lot of adenines. Protects it from being degraded in the nucleus, right? Because in the nucleus, you don't have nucleotides staying together very long unless they're being used for something. And so it allows it to make it through the distance to the nuclear envelope in order to get out into the cytoplasm. But there's a gate at the nuclear membrane, right? And so it has to be able to pass through that gate. And you can see my cute little picture here, right? So you've got your pre-mRNA, it's being processed. It's ' that poly A tail that got added during transcription. While it's being transcribed, a little cap, like a little hat, is put on the 5' end. And this little cap on the 5' end is like a pass. It's like a key to get through the nuclear membrane here. So it needs both this 3' poly A tail and the 5' cap to get through the nuclear envelope out into the cytoplasm. Now, there's a third thing that's really important, and this is called RNA splicing. So it turns out that the mRNA is actually really short compared to the coding region of the DNA, because there may be other things in that coding region that are not part of the gene, okay? So what part of the eukaryotic RNA is going to be removed from that transcript? The codons, the operons, the exons, or the introns? Well, right now, all of these words probably don't sound like anything to you. You will learn 3 out of those 4. They're all real things. You will learn 3 out of those 4 over the next couple conversations. Today, we want to focus on 2 things, exons and introns. It turns out that introns have to be removed from the transcript in order to make it messenger RNA. So the question is, why is that? Okay, exons are genes. Introns are sequences between the genes. So you may have a transcript that has a couple of exons on it, right? Well, the exons together make the gene. The introns may code for other types of RNA, ribosomal RNA, microRNAs, transfer RNA. Those get transcribed too, and they're in these introns. But in order to get the messenger RNA, you have to cut those pieces out. So these pieces get cut out, and then the exons get glued together, and this final piece is your gene. Okay, so introns will be untranslated sequences in the mRNA, and they have got to go. Exons are the translated gene sequences. This is what we make the proteins out of. And so this process is called RNA splicing. So essentially, right, ignore that alternative right there. Pretend that's not there. So in RNA splicing, we've got our DNA. In the RNA coding region, we had couple of exons, those are going to code for proteins. We had our couple of introns, they've got other stuff in them. The whole thing is transcribed. We get our poly A tail, we get our 5' cap, and then we need to cut out the introns. Those are cut out, the exons are sealed together, and now you have your mRNA. Okay. Now, here's the thing though. we just saw one gene making one protein, but there are over 100,000, maybe even as many as 150,000 proteins in the body, and only about 20,000 genes. And I'm talking about people, of course. How is this possible? We made a mistake when we identified the genes, and there are more genes in the junk DNA than we realized. We get the other proteins from food that we eat. RNA can be spliced in different ways to get different combinations of exons. DNA can be read forwards or backwards, giving you different genes. Well, that last one is just crazy talk, right? It doesn't work that way. We know that we're looking at those genes from that 5' to 3'. We know that we are transcribing them. We know we're replicating. We know we're doing that from the 3' to 5'. So there's specific directions in which we must travel. Okay, we didn't make a mistake when we identified genes, and there may be more in the junk DNA, but not 80,000 more, so that's right out. We do get proteins from food that we eat, except then we break it down to build our own proteins. So RNA can actually be spliced in multiple ways. So it makes sense, if you can cut out the introns, couldn't you cut them in slightly different ways, right? So it turns out that within these sequences there are multiple cleavage sites, right? And these sites can be used to make different versions of a single gene. And so like in this example, you have your exon, your intron, and your exon. So just normally you would cut out this intron and you would get exon 1 and exon 2 together. But you might have another cleavage site in the middle, or at the sort of beginning of exon 2. So when you cleave, what's going to happen is you're going to drop out, so this is this example, you're going to drop out that intron, but you're going to cut exon 2 before it's finished. So what you get is a different mRNA transcript. You might have another cleavage site towards the end of exon 2. In that case, what's going to happen is you're going to lose that intron, because you always will, and then you're going to have only part, but more than you had before of exon 2, but not the whole thing. And so now you have a slightly different version. This is alternative splicing. It's what allows us to take one DNA sequence and get multiple coding sequences from it. Okay, so alternative splicing, it's really, really important. And when you look at it in a nutshell, right, here's just some examples. Here's exon 1, 2, 3, 4, 5, and 6. So your transcript has all of that in it. You cut out all the introns. Normal splicing gives you just this guy. But if you can alternatively splice it, you can get different proteins, right? Here we left out 2 and 4. Here we left out 2, right? And we got 3 different versions of this gene. All right, so let's go back to just RNA processing. Let's just do the whole thing, right? The first thing we're going to do is we are going to transcribe the DNA into RNA. We begin with the promoter sequence, the transcription factors binding to the promoter sequence. Those attract the RNA polymerase, which then seats itself, opens up a transcription bubble in the beginning of the RNA coding sequence, and then it will zip along, opening that transcription bubble, and while it does, it will use the template strand to make an RNA transcript that matches the coding strand. It will eventually come to the termination sequences where the RNA polymerase will destabilize, the RNA transcript will detach. This termination sequence is after this sequence here, which is the addition of a whole series of adenines for that 3' poly A tail that will protect the RNA transcript as it is going out of the nucleus. There's also the addition of the 5' cap. That is actually going to happen while transcription is occurring. That 5' cap is a key to get out of the nucleus. Now the pre-mRNA or the RNA transcript will be spliced. It will have the introns cut out of it, right? It will have the introns removed. The introns may contain mRNA. The introns may contain rRNA or tRNA, ribosomal or transfer RNA, or other types of RNA, but those will be cut out, right? Then you will be left with your mRNA, which can then be translated into a protein, and that is a summary of what we just talked about. All right, now back to genetic expression. We know that DNA is transcribed into RNA. We're going to translate RNA into proteins. That'll be our next conversation. But the other thing that we want to keep in mind is that genetic expression is not just this part, DNA to protein, right? You have to regulate how often is the gene transcribed and translated, and how many copies do you want, right? And so there's this whole other section of a conversation about genes and how they work, and that's about the regulation of genes. Do we want more? Do we want less? When do we want it? When do we not want it? Things like that. All right, that is the end of this video notes. Thank you for listening. Please make sure to ask your questions at the beginning of class, and I will see you next time.